Why is it lethal? Why does it kill you? You got to know something about what we've been talking about, or what we've talked about with all those gradients. Oh, Michael, hold on. Anybody else? I'm going to use you as a nuclear option. Yeah. Yeah.
Okay.
Yeah.
When you inject potassium chloride with potassium gradient, you get messed up, so there would be an excess of potassium outside, so that would also mess up the sodium potassium ATPase, and then sodium would be able to get out of the cell, and other molecules would be able to be like co-transported back into the cell.
Yes. Did you all hear what she said? What she said? Yes, that's exactly right. So the bottom line is that there's a higher concentration of potassium inside the cell, right? And that potassium inside the cell is there because of the sodium potassium ATPase, right? That's what generated it in the first place. And so you're constantly transporting 2 potassiums in for 3 sodiums out. Okay, so you have to think of it in terms of what cells are most affected by this. So the whole dying part. What are the cells that are most affected? They're all affected by this, but the ones that end up killing you. So this is the normal gradient, right? So you got about 140 to 145, right, millimolar outside of sodium. Right, you got about 140 to 145 millimolar of potassium inside. Right, potassium's low outside. 4 millimolar potassium here, 14 millimolar sodium here. Okay, I inject a ton of extra potassium chloride. Right, and the potassium and the chloride dissociate. Right, so they're gonna dissociate into this. So what Marissa said is that it changes the gradient. Okay. So the first thing I want to know is which cells are the ones that we really need to understand here. All of them are affected, right? The reality is that every cell in your body is affected. True. But not every cell in the body, if their gradients are altered, will kill you. Go ahead. Neurons. So neurons may be one. What else? What else? Yep.
Cardiac muscle.
Cardiac muscle. There's another one. Here, you can say something different.
I was just going to say muscle cells.
Okay, another kind. Which specific? So skeletal, but which kind? Which one? You can help her out here. It's a skeletal muscle. Yeah. So when you change this gradient, what happens? So what's the big deal? Everything's been said is correct. What's the big deal that there's such a high amount of potassium outside the cell? Isn't it being transported into the cell anyway? So what's the big deal? How come these are affected? Neurons, cardiac muscle, and the diaphragm. What's the big deal? It's just a gradient. What can't happen? Well, right. So there's these leaky potassium channels, right, that constantly leaking out. So that's one problem. Okay, because they can't, because you'll actually have potassium coming in because they're leaky channels. They'll move potassium in either direction. They're just open. That's one problem. What's another problem? This is at rest. These potassium channels leak all the time. What about when these 3 are active? What's going on? Yep, here.
After the action potential to repolarize the cell, you have to let the potassium back. Wait. Sorry, I'm getting.
That's okay. You're right. So far, you're right.
Back into the cell, or you let it out of the cell.
And what does that do?
But to repolarize the cell so that you can bring it, polarize it so that you can generate another action potential. But if there's a high amount of potassium outside of the cell already, it's not going to want to move up its concentration gradient.
That's correct. So potassium chloride, there are a bunch of solutions that we use even in research to sacrifice animals, like sodium pentobarbital and things like that. And a lot of them are based, euthanasia, right, to sacrifice animals, are based on potassium chloride. And this is very simple principle. So if you go into healthcare, you know, when there are different types of bags, right, that you can give somebody for an IV. This is usually 1,000 mils. This one's usually like 250 mils. So the bags are different sizes. And this one sometimes has dextrose in it, which is basically glucose, right? And it's got 1,000 mils, and the osmolarity here is, I don't know, somewhere between 290 and 310 milliosmoles per liter, right? This is not a big deal. This will just make you swollen, right? You retain water. This one is in a different bag. It's in a different size for a reason, okay? Because one, you don't. This is the one that's lethal. If you give it at the wrong time to the wrong person in the wrong dose. And the drip rate, because you can set the drip rate here. There's a piece of plastic here that allows you to set the drip rate, and you can set it per second. Okay, and so even the drip rate makes a difference. So when you go into healthcare, when I taught some nurses, I would always make a joke of it, but this is a big deal, right? Like the difference between potassium chloride and understanding why potassium chloride and sodium chloride are so different is the difference between life and death, essentially, right? Because even if you give injections, you can buy potassium chloride pills. Same thing. Now, there is a dearth, a lack of potassium being consumed by Americans because of the way our diet is, right? So it's actually a bigger problem than sodium, but it's unlikely that you're able to eat enough potassium to cause this phenomenon to occur. Does that make sense? Potassium is good, right? You should probably eat more potassium than you eat sodium chloride, but to eat this amount, it's just not very easy to do. But to inject it, or if you're taking it in pill form, which you can do, that would be problematic. So this is sort of a big deal, and the understanding of the why it's a big deal, the how it happens, to me is the most important thing. You could have looked it up on the internet and say, well, it'll kill you. And that's great. Of course, it's going to kill you, but why? Okay, so hopefully that's everybody gets that because it's super important. Okay, 3 most important things last time. What did we do that was really important? Anything I told you to memorize? Yeah. What did I tell you to memorize? Yeah. Parts of the sarcomere, hierarchy. What else? What else is important? Yeah. Cross-bridge cycling, how that occurs. One more. So one more, and then I'm gonna have to borrow something from somebody. Somebody that doesn't usually say something. Just say something, anything. Bueller. Titan. It's a spring-loaded, right? And Titan is not. What's that? It's the biggest protein in the body, and how far does Titan extend in the sarcomere? From what to what? Remember? It starts at the Z disc and it goes to the help mount. How long is Titan? It's the largest protein in the body. It's like a spring, but how long is it? It starts at the Z disc. M line. That's correct. It connects everything together. Yeah. Okay, so if I may borrow, there's a handout that I gave you on excitation, contraction, coupling, and I want to take you through it. Right? I want to explain it. Yes. guess, if I remember what I'm saying. Okay, so it looks like this. We're going to start in the very left-hand column. Now, some of this is going to resonate with you because we've sort of done parts of this in neuro and now in muscle. Now I'm going to try to put the whole thing together. So, if you start in the very left-hand column, number 1, it says skeletal muscle voluntary. The stimulus is you think about a movement. Now, between think about a movement and a motor neuron, I just want to make sure that you all realize that there's a lot of steps that I have skipped here, right? Because of all the connections. I'm not going to ask you to give me all of the pieces in between because, and that would go to that diagram where we looked at the parts of the brain that are involved in this. So we're going to skip that part. But I want you to at least have in your mind that where is this thinking going on? It's going on here in the prefrontal cortex and the frontal cortex area, and then it's tracking its way back and then down. So we're just skipping it. So we're going straight from that to a motor neuron, right? So a motor neuron will generate an action potential. Did you have a question?
No.
Okay. A motor neuron generates an action potential, and we've looked at that diagram multiple times at this point, right? So everybody knows the diagram, should be able to draw that diagram. So you could draw this, an arrow from prefrontal, frontal cortex, higher brain areas to the motor neuron if you wanted to. And on an answer to an exam, you could say many steps have been skipped here. There are many areas that might be involved. That's fine. Just I want you to be aware that there are parts in between. So the motor neuron generates an action potential, right? You know what an action potential is. You could draw a graph of a general action potential. These muscle cells, again, these motor neurons have action potentials between -40 and -70, give or take, right? The action potential, remember, is an electrical signal that travels down. And that electrical signal current, the actual movement, right, the actual electricity is current. And in the human body, that current is positively charged. So it's what's coming out of the outlets here, outlets here, is a negative, right? Because in metal, the metal particles don't move. The only thing that can move in the metal are the electrons, and electrons have a negative charge. But in the human body, remember we were looking at saltatory conduction with the myelin sheath? Why are we moving positive current? Because that's what we're moving. Why is it positive current and not? It could be negative too, but it's mainly positive. What is moving in and out of the cell that's generating this positive current? Sodium and chloride are both positively charged, right? So it's that movement with those channels. Remember, at the nodes of Ranvier, there is a huge density of sodium and potassium channels. And that's why the signal gets sped up along the way. All right. So it comes down the axon. So we get to step 3. It comes down the axon, and you open up N-type, N as in Nicholas, type calcium channels. And they're called fast calcium channels. So those channels, the N-type fast calcium channels, have a voltage sensor built into them, right? So they're voltage sensitive. And you know what voltage is, right? It's the membrane potential. It's the potential. So they have a sensor in it. So when the current comes down, it changes the membrane potential, right? And this calcium channel senses that, and it allows, now here's what we didn't look at. What it does is it allows, and here's the bouton, right? It opens up these calcium channels, and calcium comes in to the actual axon. Why does calcium come into the axon when I open up this channel? It's down, it's coming down, it's electrochemical gradient. That has to be the answer, right? Or this isn't a channel at all, and this is an ATPase, and that's a different beast. And then you know exactly what's happening there, and there it's the reverse. It's going up its chemical gradient, right? Electrochemical gradient. But that's not the way we do it. We set up these gradients in your body because they're potential energy. Remember, they're motors that we can use. All right, so calcium then enters the axons. All right, so in between here, number 5. Acetylcholine-containing vesicles fused to the synaptic membrane. Now, in this, when I wrote this years ago, we didn't have a book that was this good, and we didn't have this whole neuro section, and so I skipped steps, right? But you know more than this. Why do the vesicles that are in here, floating around, why do they fuse to the membrane? What does calcium do to them? Yeah, Daniel. Synaptotagmin, right? So they bind synaptotagmin, which is here. And what does it do to the synaptotagmin in the vesicle? What does the calcium physically do to it? Is the synaptotagmin in the vesicle the same with calcium bound as calcium unbound? It is not. That's correct. So what's different about it other than the calcium being bound? What does calcium do? Let me give you another question. What does calcium do when it binds to troponin? Go ahead. Yeah, there's a conformational change. It changes shape, and that shape change causes an attraction to the membrane. And then this thing here, remember, is a snare protein. And the snare protein is what attaches to the membrane itself. Right? So this is the calcium works like an on switch. It flips the switch, the lights go on, and now this can be attached. So that's missing here. But I went over it, so I suspect and expect that you would know that, right? Because I did go over it, and we've gone through it many times, even though it's not on the sheet. Because this is an old sheet. Okay. Does everybody understand that? So then there's a fusion, right? Acetylcholine is released via exocytosis into the synaptic cleft. We've talked about that before. We've seen that many times. Number 7, acetylcholine binds to the nicotinic acetylcholine receptor. That's really, really important, right? Because there are 2 major types of receptors. It's not a muscarinic receptor here, right? That wouldn't do the same thing. It can't. It's a nicotinic, and it's at the neuromuscular junction. So I'm not going to draw the whole thing again. The nicotinic acetylcholine receptor is an ionotrope, right? So that receptor, remember, when we looked at types of receptors, the first type of plasma membrane receptor, right? If this is the receptor, it is attached to an ion channel. So this is acetylcholine. Here's the receptor, and it opens up an ion channel. And this ion channel is a non-specific sodium channel. Okay? Meaning that if an ion is smaller than sodium, it'll make that one come through too. But it's mainly sodium. Okay? So the number 8, nicotinic acetylcholine receptors are ion channels that conduct sodium and potassium, causing a transient end plate potential. Now, we went through end plate potential, right? You notice how all of this is starting to come back, like all of the pieces are starting to get put together? So what does that mean? This causes, this going through here, remember, this is happening down here. When this comes through, it causes end plate potential. What does that mean? An end plate potential. Do you know what it's a type of what? So the first question you have to ask yourself, what is an end plate potential? What type of potential is it? So it's in a class. There are 2 major classes of potentials. One of them is an action potential. The other one's a what? Graded potential. So it's a type of graded potential. So What are the characteristics of a graded potential? The most important characteristic of the graded potential. What's that?
So it's decremental.
It's decrementing. So if this is the point where you generate this thing, what's going to happen to the current? What's going to happen to the potential as it goes from here and here out? Decrementing. It's going to decrease. That's right. So you're going to get a, we'll note this like positive charge here, right? And what we'll do is we'll make 3 of them come here, 2, 3, 2, and then by the time you get out to here, there's no voltage change. Okay? Did everybody get that? So this is only the first step in sodium channels that are opening. Just like this is the first time you see calcium. You're going to see the sodium channels open up again. So does anybody not understand that? Does that make sense to everybody? And you remember, where are these neuromuscular junctions located in the muscle fiber? So along the length, where are they located? If you had positioned them. If you had a muscle fiber, and I say, tell me where the neuromuscular junction is going to occur. The middle, right here, which totally makes sense based on the end plate potentials. Okay, so if this is decrementing, right, it's going to start from the middle. Okay, start from the middle. All right. So we're going to go to step 9, which is on the back. Step 9 says that the end plate potential causes voltage-gated sodium channels to open, causing a huge influx of sodium to enter the cell. Didn't we just have sodium enter the cell here? Yes. Is this a voltage-gated sodium channel? No. It's not. It's an ionotrope, right? So this is an acetylcholine ion receptor, a nicotinic acetylcholine ion receptor channel. Down here, somewhere in the range of the end plate potential, on both sides, you actually have sodium channels that are fast sodium channels. And they're voltage-gated fast sodium channels. So these are voltage-gated fast sodium channels. So when this thing creates an end plate potential, right? When this ion here, this positive ion hits the voltage sensor opens the channel, and you get a huge influx of sodium. And this one will not make potassium come through it. Here, potassium can sneak in. This one will not. They're different. Okay, this is voltage-gated. This is an ionotrope. This only allows a little bit of sodium to come in, just enough to generate this end plate potential. These then generate massive, and what ends up happening is that these are the ones that are going to regenerate the action potential. Okay? These are the ones that are going to regenerate the action potential. This one does not. This one generates an end plate potential. These regenerate an action potential. Does that make sense to everybody? That does understand that there's 2 sets of sodium channels you're talking about, right? This one is associated with acetylcholine, this one is not. This is the first time you see calcium. We're going to see calcium again. But the calcium we're going to see is going to be on this side now. All right. So this causes depolarization. So if you had to draw depolarization, you could do that, right? You can draw the sodium coming in, right? It goes up to plus 20, whatever it is, and then it's going to have to repolarize. Okay. the depolarization propagates down T tubules. All right, let me see if I have a picture of this. Okay, we do. If you could, well, I'll go. So I don't have to draw this. So this is a muscle cell, right? And in there, you see in here there are 5 myofibrils. Okay, so sarcoplasmic reticulum. So these are the myofibrils. That sodium channel generates that action potential. That action potential, positive charges come down these T tubules. This is called a triad, right? You have the sarcoplasmic reticulum, which is a specialized type of endoplasmic reticulum that flank these T tubules. The T tubules sit on the outside of the cell. This thing is a muscle cell. These are myofibrils, right? These are not muscle cells. This whole thing is a muscle cell, right? So these T tubules are on the outside of these myofibrils. So this positive charge is going to come down each of these T tubules. That's what the T tubules. So they're an invagination that conduct current. They're specialized to conduct current. Okay, so like a funnel, like a river. So what these two generate will come down the T tubules. So think about the T tubules being underneath them. And the current, positive charge, will flow down that T tubule. Okay. Does everybody get that? Does that make sense to everyone? If it doesn't, you can tell me. It's time to fix it now. All right. All right. Step number 12. It says the propagation continues until the terminal cisternae. The terminal cisternae are these SRs, the terminal ones. So these sarcoplasmic reticulum, they're all along the muscle fiber. Okay? So these channels, they conduct a current that goes out this way and then ultimately goes down, right? Because it's an action potential. It propagates through the entire muscle. It will come down these T tubules, right? And then somewhere around here, somewhere over here, over here, on each of these myofibrils, so there'd be one here, and one here, you have another voltage sensor. Okay, you have another voltage sensor, and that voltage sensor is called DHP, or dihydropyridine. Lots of voltage sensors. So that dihydropyridine, and I will try to draw this. That dihydropyridine so if this is the T tubule, right, the invagination, you have this current coming down the T tubule over here somewhere, like this, and I just draw it as a box, is this DHP. And when the current passes by the DHP, it turns on the dihydropyridine. Okay? It senses voltage. Now, that dihydropyridine is also connected to something called a ryanodine receptor. Receptor. So when this thing turns on, it turns on the ryanodine receptor. Okay? So the ryanodine receptor is not sensing the current in the T tubule, the DHP is. The DHP, then, there's a bunch of chemical reactions that occur. You don't have to know them, right? Very cell biology, but that activate the ryanodine receptor. Now, the ryanodine receptor is attached to this thing. All right, so let's see where we're at. We'll talk about that in a second. So DHP is activated. This voltage change is then transmitted from DHP receptor to the ryanodine receptor of the sarcoplasmic reticulum. This is the sarcoplasmic reticulum. Where do you see sarcoplasmic reticulum here? You see this mesh netting here? All this mesh netting, that's the SR. I draw it as a big old box because I can't draw it. But it's not a big old box. It's just easier for me to visualize it that way. But this, all of this mesh netting is the sarcoplasmic reticulum. Any questions so far? All right. So calcium is released from the SR. That ryanodine receptor is essentially a calcium channel. So that ryanodine receptor, right, because of it's turned on, is a calcium channel. So it will then cause this to open and calcium is released. Now, it is not an ATPase. I have not told you anything about the SR other than it's part of the muscle. Why does the calcium leave the SR? What has to be true if it's not an ATPase? It's down its electrochemical gradient. So what does that mean? What is this? What is going on in here? That's right. It's down its electrochemical gradient. What is going on in here? What does that mean? Yeah, the density of calcium here is super high. This thing is jam-packed with calcium. It has to be, because if it's not, you're going to open up a gate here. It's not going to flow out. It can't, right? Okay, let's keep going here. So calcium then binds to troponin. Now, mind you, all of this is going on. The sarcoplasmic reticulum, right, is attached to the myofibril. What is a myofibril made up of? What did I show you that you put them back to back? What do you put back to back and make a myofibril? With elephants, giraffes, flying monkeys from the Wizard of Oz. What is this? Sarcomeres. That's right. Sarcomeres. Okay? So this thing here, right, is, you can think about it, it is sitting over, right? I'm going to do this in a different color. This gets a little complicated. So this is a myofibril, right? A myofibril is a type of what in this cell? This is a myofibril. It is a type of what? What lives inside cells? Say it again. Organelles. It's a type of organelle. That's what it is. It's in the cell. So there's a big old cell here. Right? Big old cell. This is just one myofibril. Calcium gets released. This myofibril is made up of, let's say it's made up of 3 sarcomeres. Right? It's made up of 3 sarcomeres. The way I draw my sarcomeres, and this is overly simplifying this, right? Right? And then same thing here. Actin and myosin. Everybody with me so far? Okay. So, it says in step 15, calcium binds to troponin. Okay. What are these two things here? Which one's actin? This one? Yes. So this is actin. Correct. So then the other one must be Myosin, correct. Okay. It says in step 15 that calcium binds to troponin C. The next question is, where is troponin C located? In this sarcomere. Where is it located in the sarcomere? Yep. Oh, you had a 50-50 chance of getting it right. It's not located on myosin. So where's troponin C located? Actin. That's correct. So it's here. So if I take this, right, and I blow this thing up, I'm going to blow this thing up for you, right? What this thing actually looks like is this. Right? That's what that looks like, right? Isn't that what we saw the other day? These are G-actin molecules. The whole bead of them is called, the whole chain is called F-actin. The blue line is called tropomyosin, right? That covers these active sites. And the troponin complex is attached. So what we're saying is that calcium comes in and binds to this. It binds there. Somebody take me to the next step. When calcium binds to that troponin C molecule, what happens? Tropomyosin moves off the active site because there is a conformational change made in the troponin-tropomyosin complex. Yes. So it moves off of the active sites, so we're going to move it off of the active sites by the magic of animation. It's kind of like watching a Disney movie. Not really. Okay. So it moves off, partly. Okay. Now, let's go to the next step. Number 16. If the troponin T, which is attached to tropomyosin, pulls the tropomyosin away from the G-actin binding sites, while troponin, misspelled, is attached to the F-actin filaments. Okay? That happens. Now, we're in here. What do we need to draw? Well, and these are doubles. Okay? What are these things that I just drew? Myosin heads, the light chains. There's two of them. The double golf clubs, remember this is the heavy chain and it's wound, right? Because this actually looks like a snake. That's what it actually looks like. Okay, now that those sites are exposed, what are those heads going to do? Bind. They're going to bind. So these are going to attach to these active sites. And as soon as they attach, that's called a what? Remember the term? It's a goofy term because it has two meanings. Cross bridge. That's right. And as soon as a cross bridge is formed, what has to happen in muscle? Contraction or ratchet. Towards the middle. Towards the middle, right? So things are going to move in this direction. Okay? Why is that possible? Why is that possible? How can that ratcheting, that contraction occur? What had to have happened long ago to that head? What kind of position is it in before the attachment? Say it again. It's cocked back. So what made it get there? Because remember, cocking back takes energy. It's not a simple thing. You're not moving something down the concentration gradient. So what had to have been involved? ATP that got it there. That's correct. And then as soon as it's as soon as this attaches, you get a ratcheting towards the middle. You get the same thing happening here, and the muscle shortens. Okay. Yeah.
Are there different kinds of troponin? Is that like troponin C, T?
Yes. Yes. So this is C, this is T, and this is I here. The I is attached to actin, the T is attached to tropomyosin, and the C is attached to calcium. Because there's 3 pieces. Yep. Okay, so in step 17 it says the light chain myosin ADP heads bind G-actin, forming cross bridges. It causes a power stroke spontaneously, causing a muscle contraction. Okay, everybody with me so far? Got it? Okay. Now we got to relax the whole thing. So you got to basically go in the reverse direction for the most part, not exactly. So in step 18, ATP will bind to these heads, right? The light chain myosin head, therefore decreasing the affinity of the globular myosin head, the light chain, for G-actin. The binding of ATP causes the heads to separate from the myosin. As soon as that separation occurs, this head is also an enzyme. Okay? It's also an enzyme. It's an ATPase. Right? What does ATPase do to ATP? It's an enzyme. Yeah. Yeah, that's right. So that it converts ATP into ADP plus PI. That's what an ATPase does. Now, we use it in the terms of the sodium potassium ATPase, which is a pump, which has this enzyme attached to it. But that enzyme can be attached to anything. It doesn't have to be attached to a pump. It's not attached to a pump here. It cleaves, it hydrolyzes ATP. So the terminal, right, the terminal phosphate bond is broken, and that's a high energy bond. Okay. ATP is hydrolyzed, causing the storing of energy in the myosin head for the next power stroke. It cocks the head. It moves it back, and that takes a lot of force. It takes energy. All right. The G-actin molecule is again covered by tropomyosin. So the other thing that has to happen here is that calcium has to dissociate from this, right? Calcium doesn't stay bound forever. There's a time limit. There's a clock that ticks on how long calcium will stay bound there. Does that make sense? If calcium continues to stay bound, let's say you have this problem. Let's say you go through all of this with the ATP to ADP, and the calcium, you've engineered this calcium and troponin C molecule to be linked forever. They're terminally linked. What's going to continue happening here? You say it. Contraction. You won't be able to get out of this contracted state. Right? You'll be able to get out of this part of the contracted state. But as soon as these two dissociate, the whole thing starts over again. Right? Because the tropomyosin never ends up covering those G-actin binding sites, because they can't. Because the calcium keeps it in a conformational change that is open. That's a problem. Okay? It's not a problem most of you have, but it is a problem. Okay. ATP is high. The G-actin is covered. So when that happens, the calcium comes off. The tropomyosin comes back and covers them up. Calcium is resequestered. There's a new word for you. Resequestered by the sarcoplasmic reticulum. Now, the sarcoplasmic reticulum is here. So there is a protein that resequesters calcium, brings it back in. So this thing has to shut down too, right? This thing has to shut down. This thing has to shut down. How does the calcium get back into this? What kind of a protein is this that allows the resequestration of calcium? Based on what you know, you told me about how it left. How is it going to get back in? Daniel? It has to be transported. Yes, by what's special about this transporter? A couple of things. It uses AT. So this also has an ATPase, right? Attached to it. It has to. And this thing will only transport calcium back in. So it's pushing it uphill. And this is a really big hill, by the way. This is a mountain. This is Everest. That's how big of a hill. Because that's how much calcium you can pack in this thing. They may be small, but they pack a wallop. So calcium sequesters. This pump is called, it's CIRCA for short. The sarcoplasmic endoplasmic reticulum calcium ATPase. That's a mouthful. CIRCA. Now, once you get the calcium in there, you then need a mechanism to pack it in. Right? Pack it in. So if you've ever, done, we all do stupid things in our lives when we're young, right? If you've ever been in a hotel with an athletic team or band or whatever, and you're young, you're not necessarily drunk because you're too young to be drinking, and if you've ever done this, right, like, there's an elevator. Let's see how many people we can jam into this thing. And then you get in the elevator, and 20 people get on the elevator, and then the elevator goes up, and you realize that the bottom of the elevator and the floor don't match up by about 7 inches. You've never done this before? I didn't tell you, don't do this. You've never heard this before. You've never done this before? Okay, well, maybe my generation did stupid things like that. But when you do it, you have to step up in order to get out of the elevator because it's rated at 2,000 pounds and you have 3,500 pounds in there. You won't die, but you might break it. Not a good idea. Anyway, so let's say you did that. You got to pack people in, right? It takes energy. People on top of people. Well, put her over there because she's smaller. Well, you're bigger. Why don't you go in that corner? We can get a few more in here, right? Just jam them up, throw them up on top. Yeah, right? So this is what's happening. It's an elevator, and they're having a party. So the coordinator of this party is a protein called calsequestrin. Calsequestrin is like a sponge, and it allows more calcium to be stored in that SR than should normally be capable of being stored in there. It basically packs the molecules in there really, really tightly. So that's why the gradient is huge here, because you need that for muscle contraction. You need this really large gradient. Okay? So that's what calsequestrin does. It's like a sponge. Okay. Then, way back at the neuromuscular junction, remember this thing? Way back at the neuromuscular junction, remember that there was here, which is here, something has got to bust this acetylcholine off. And there's an enzyme called acetylcholinesterase. And acetylcholinesterase breaks this thing into two molecules. Acetate plus choline. And this comes off, so that stops this part of the contraction. Okay? And then remember, that has to be reabsorbed. And so that stops the positive charge, that stops the end plate potential. All of this stuff that's happening here goes away. All right. That's excitation, contraction, coupling in skeletal muscle. Any questions on that? Because what I'm going to do real quick, I'm going to go through the cardiac today. On another day, I'm going to take you quick through the nuances, and I think it's actually in the presentation, of the difference between these two and smooth muscle. So if you understand that, and if you don't, I need to know now. Not tomorrow, not next week, not the day before the exam. It's too late. If there's something I said that you don't understand, I got to fix it now. Tomorrow's too late. Okay? So is there anything that I've said that's a little confusing? I'm drawing lots of pictures, right? I wish there was a way to sort of bring this thing and do it all in one fell swoop. This is about as close as I can get. So you have the paper, you have a bunch of these different diagrams that sort of come together. Is there anything in here that doesn't make sense, that's confusing to you on how this process works? I mean, not memorize, having all the steps memorized, that's okay. I don't expect that at this point. right? But just you understand conceptually what's happening when I go from step to step to step. If you get that, then the memorization is not that hard. If you don't get that, you're going to memorize garbage, right? Because you're not going to even know what you're memorizing. And the likelihood that you're going to remember something that you don't understand is not very good, right? So you have to understand. So does anybody has any problems with any of this? Yeah.
I just have a question. What is the blue positive?
The blue positive
What
here,
is that like?
that is the current coming down through the voltage-gated sodium channels into the T-tubules. So that, remember that T-tubule is like a river bed. In this case, it's not water, it's current, positive charge that comes down it, right? And it's insulated. So that the only piece of protein here that can respond to this is this DHP receptor, which has part of it in the tubule. Everything else, there's no current leaking here, right? This current doesn't leak out this way. It can't, it's insulated. So you funnel it down, because if you were to do that, you'd have the same problem with an unmyelinated axon, right? Which is not insulated. You'd leak current, which would slow down the transmission. And you don't want to leak current. So this whole thing is insulated. Only when it gets to here does this thing respond. Good question. Other questions? There is good? Okay. If you understand that, we're going to just take you through the nuances now. Good. I just turned my lights to it. The nuances between that and cardiac muscle. Okay? Cardiac muscle. They're very, very similar. They both have sarcomeres. The sarcomeres, they're both striated, right? If you look at that list of differences and similarities between the muscle. So let's talk about just the differences. And then when you study this, when you look at this, have some more time, maybe then you'll have more questions because you'll be like, well, now I don't understand the connection. All right. The very first step is different. In your heart, you have a pacemaker called the SA node, right? The SA node has what are called F-type, and it's not on here, F as in funny, because that's what they're actually called. They're called funny sodium channels, and they leak sodium. They're one of the few leaky sodium channels in the body. Lots of leaky potassium channels. But this is specific to the heart. There are some others. Okay, so you have leaky sodium channels. That's the trigger for the heart to start this entire process. That started in your skeletal muscle by you thinking about it. Do you think about making your heart beat? You can't. I mean, you can do it, but it's not going to help. Like on, off, stop. You can't do that, right? You can modulate heart rhythm. You could speed it up or you can slow it down, and that does come from your brain. That actually does come from the parasympathetic nervous system, the sympathetic nervous system, right? But you can't actually start or stop this process. It'd be nice if you could. It's not a good idea. It's the whole stopping doesn't work really well for most people. Yeah. So, just in case you weren't aware of that. All right. So, everything else from step 1 down through the activation to 11 is the same. The relaxation is now different, slightly. Okay, where is the skeletal muscle getting all of its calcium from for contraction? I haven't said this before, but you can make this leap of faith based on everything I've told you. Where is a skeletal muscle cell getting all of its calcium for contraction? What's the structure? Yeah, the SR. Okay? The SR. Now, there are some other slight differences. Okay, before we get to relaxation. The cardiac muscle, the SR in a cardiac muscle is slightly smaller. So, in the cardiac muscle, it's getting most... All right, we're going to have to pull those out. Sorry. It's getting most of its calcium from contraction from outside the cell. So there are calcium channels that open up. So in a skeletal muscle, it gets all of its calcium for contraction from this structure. In the heart, it gets most of its calcium for contraction from extracellular fluid. Remember, this is intracellular fluid. And only some from the SR. Why can it do that? What are the gradients for calcium inside and outside the cell? Give or take. It's like 2.5 millimolar outside of the cell. What's it inside the cell? 10 to the negative 4. 1 times 10 to the negative 4. So there's a huge gradient there, right? So when this process happens, not only do you have this occur, but remember that along the muscle membrane, this thing going on here, remember that an action potential will travel very, very far. That this is a voltage-gated calcium channel. Only it's an L type. It's not an N type. It's slower. So the current comes along here, and there's a voltage sensor in here which opens up this channel at the same time that DHP turns on ryanodine to make this channel work. That's another difference in the heart. You don't have these channels in skeletal muscle. They don't exist. Does that make sense to everybody? So it's slightly different. Okay. And then on the way out, so that was on the way in. That was a nuance on the way in. On the way out, all of this part stays the same. So all of these pieces here are the same. But anticipate what has to happen. What do you have to have here in order to get the calcium that came in from outside the cell? You don't have to know exactly. Generally speaking, what has to be here in this plasma membrane? Well, there's two. I'll skip through the chase because we're getting late here. You actually have a calcium ATPase, which is a pump, and then you have a transporter, which we've already looked at, that's called the sodium calcium exchanger. So these two are going to get the calcium going out. This one, because it's going against its gradient, this one uses sodium coming down its gradient, and when calcium binds, it gets shot out against its gradient. So this one is secondary active transport. That's a nuance. Okay? That's about it. All right, real quick, because we're a little bit over on time here. Just look at this. Try to understand this. Definitely look at the smooth muscle one, too. Next time, we're going to finish up muscle here, and then we'll also talk about smooth muscle, which is a little bit different. Okay, important things. Maybe it's not even pieces of information. Maybe it's advice for your friend who is sitting next to you and dozing off. What would that? 3 things. It doesn't just have to be facts. It can be a piece of advice for someone sitting next to you around the corner, because you feel sorry for this person because they're just not very good students. Because anything? Yeah. Yeah. Look at the sheet and make sure you understand from step to step. Number 2, read my mind. Yeah. We have a quiz on Monday. Very important. Remember that. See? That didn't come in the lecture. Number 3. Memorize the steps. Okay? I'll do number 3 today. Have a great weekend. Oh, yeah, I'm sorry. That's right. I do have a question. Right. It would be challenging.